Welcome everyone. We're going to be looking at gene expression. Gene expression may sound a little bit weird, but it's exactly what it sounds like. If you remember, your DNA contains many genes on them. Each gene can code for different things. So for instance, you can have a gene that codes for your eye color. So the way that the gene is going to become the protein that actually gives your eye that color is known as gene expression. It is converting that DNA language into the proteins that actually make the physical appearance. So in order to follow through the steps and learn how DNA is converted into protein, we first need to understand some basic things. RNA. RNA is going to be really important. So far, we've talked a lot about DNA, and we saw how DNA replicates and why DNA matters. Well, RNA and DNA are both types of nucleic acids. So going back to your biological macromolecules, you should remember that you have 4 major biological macromolecules: carbohydrates, proteins, your lipids, which can include your fats and your oils, and then your nucleic acids. DNA and RNA are both types of nucleic acids, and that means that they're made up of nucleotides. And if you remember, each nucleotide is going to be made up of a phosphate group, a sugar, and a nitrogenous base. In the case of DNA, those nitrogenous bases are going to be A, T, G, and C. You already saw this when you looked at DNA replication, because when DNA is always going to be in a helix, right? And it's always going to have complementary strands. So A with T, G with C, and so forth. A will never pair with G, C will never pair with T. It is always A with T and G with C. RNA is going to be different in several ways. One, it's going to be single-stranded. So while your DNA has 2 strands, your RNA would only have 1 strand. While your DNA has 4 types of nucleotides, A, T, G, and C, RNA is going to have a U, G, and C. So here the difference is, instead of having thymine, you have a nucleotide called uracil. Okay, and we'll look at that in more detail. The next is going to be that DNA has a sugar called deoxyribose, which is where it gets its name from. So DNA is deoxyribose nucleic acid. RNA has a different type of sugar called ribose. So RNA is ribose nucleic acid. The D from DNA comes from the type of sugar. The R from RNA comes from its type of sugar. Please know these 3 major differences between your DNA and your RNA. RNA is going to be key in making proteins, but of course, it's going to be key only because it's derived from your DNA. And so you can see here, this is a DNA molecule. This is an RNA molecule. The RNA molecule is going to be made from the DNA molecule using complementary bases. And we're going to see how this works. So gene expression, getting from DNA to protein, is a 2-step process. The first step is going to be something called transcription, and this takes place in the nucleus, as you can see in this image here. The next step is translation, which takes place on a ribosome, and that ribosome can either be free-flowing in the cytoplasm or it can be attached to the rough endoplasmic reticulum. So the process of converting that DNA into a RNA molecule into a single-stranded molecule is known as your transcription, and it occurs in the nucleus. That RNA molecule will then exit the nucleus, attached to a ribosome, where it will be read and make a protein, and that process is known as translation. Please know where each of these 2 key steps takes place in the cell, and know exactly what they do. That one converts DNA into RNA, and that the other one converts RNA into the protein. Right. Those are your 2 key steps in genetic expression. Here's another image showing that. In this image, you're looking at a woman. As you know, she is composed of billions of cells. Each cell contains chromosomes. In humans, we have 46 chromosomes in every single one of our cells, if you remember, except for our sex cells, in her case, her ovaries. The eggs within her ovaries will only contain 23. All the other cells of her body contain 46 nice little chromosomes hanging out. Those chromosomes are made out of DNA, which is in a double helix. When you're going to make a protein, in this case, all of these different proteins that make her up. She has brown hair, she has brown eyes, she has type AB blood, she is lactose tolerant, she has normal blood cholesterol. All of these are different proteins that are coded on genes within the DNA. Those genes need to be converted into an mRNA strand. The reason they need to be converted into an mRNA strand is because DNA is too large to exit the nucleus. So if you remember from when we looked at plasma membranes earlier in the semester, you know that certain things can easily pass through the plasma membrane because they're small enough or because they're in favor of their concentration gradient. In the case of DNA, it is just not small enough to fit through the pores of the nuclear membrane. So DNA is only going to be contained within the nucleus. RNA is small enough to exit the nucleus. So when you make RNA, there's a lot of RNA then in the nucleus, and it will freely exit the nucleus and head to the cytosol or cytoplasm, where it will encounter a ribosome. That RNA is then going to be converted into a protein, and that protein can code for being lactose tolerant or intolerant, having brown eyes, green eyes, blue eyes, hazel eyes, brown hair, red hair, blonde hair, your type of blood, whether you have normal cholesterol or a different type of cholesterol. This is your genetic expression. All right, so let's start with step 1, transcription. So again, remembering that to get from DNA to RNA to protein, you have to go through two steps. Step 1 is moving from DNA to RNA, known as transcription. Step 2 is moving from RNA to protein, known as translation. We're going to start by talking about the first step, transcription. In transcription, you're going to take that DNA molecule and you're going to convert it into an RNA strand, specifically something called mRNA, because the M in mRNA stands for messenger. It's the messenger molecule. So this RNA you're making will then exit the nucleus, find a ribosome, and then be converted into a protein. That's why it's called a messenger RNA, because it's carrying the message from DNA to the ribosome to make a protein. To make your RNA molecule from your DNA strand, you need to follow the base pairing rule. So just like when you have two DNA strands, you can have A with T, G, sorry, that came out badly, G with C, C with G, T with A, etc., etc. These are your two strands. One of them will code for the new RNA molecule. The other one is going to stay the way it is. So the one that's going to code for the new RNA molecule is called your template strand. The other one is your complementary strand. In this case, I'm going to make this one here be my complementary strand, the one that I just highlighted in yellow. That didn't work really well. Let's highlight it in a green. Okay, this one here is going to be my my template strand. The other one is going to be my complementary strand. And I'm going to make an RNA molecule from this. So following the base pair rules, if I have a T, that will connect with an A. If I have a C, that will connect with a G. If I have a G, that will connect with a C. And then if I have an A, that should connect with a T. But because I'm making RNA, remember that RNA, one of its differences is it does not have a T. It does not have thymine. Instead, it has U, which is uracil. So your template strands will make AGCU, which is similar to your complementary strands of the DNA molecule. See how this guy here and this guy here are almost the same, except for wherever there's a T, now there's a U, because they're both pairing to that template strand. And then this guy will be your mRNA strand that is being made, and then we'll enter the nucleus. RNA polymerase is the enzyme that is key in conducting transcription. It's the one that's going to help unwind that DNA molecule so that the template strand could then attach the complementary base pairs for the RNA strand, and RNA polymerase is going to be key in that. There are 3 steps that take place during transcription. Please note that these steps are the same as in translation, except for they're going to do a little bit of small differences. But all 3 steps are going to be called IET: initiation, elongation, and then termination. In initiation, there is something called a data box. So that DNA strand is going to contain something called a data box, which is where the RNA polymerase will bind to start to initiate the process of transcription. And at that point is where DNA molecule will start to unwind so that only one strand, the template strand, is going to be the one from which the RNA molecule will be made. And then your RNA synthesis starts. And that synthesis is the adding of nucleotides to that DNA molecule. So you're adding nucleotides, but remember that you're going to be adding these guys over here, the complementary RNA molecules. Elongation is the process of continuously adding those nucleotide bases until you reach the end of that gene. Okay, and the RNA polymerase will always read in a 5-3 direction. This is similar to what you looked at when you're looking at DNA synthesis. Remember that there is a 5-3 and a 3-5 direction on DNA, and reading is done in one direction. RNA polymerase always reads in the 5-3 direction as it goes adding those complementary RNA nucleotides. And then finally is termination. And this is where you are going to reach the end point of that gene. The RNA polymerase will then detach. Remember that it attached during initiation. It will now detach from the DNA molecule. And once it detaches, the RNA transcript molecule separates from the DNA molecule, and it is a full RNA molecule. And voila, you just made RNA from DNA in 3 little steps: initiation, elongation, and termination. But that's not it. Before the RNA can actually exit the nucleus to the cytoplasm, it's going to have to do a few things. And we're going to look at that in just a moment. Here, figure 14.8 of your book does a good job of explaining these 3 processes of initiation, elongation, and termination in a little bit more detail. And you can see here the initiation. This is the start point where the RNA polymerase will attach and read in the 5-3 direction. The DNA unwinds. It goes reading and adding nucleotide bases. This is your elongation. And then once it's reached the end of the gene, the DNA polymerase will detach. The DNA will rewind, and your RNA is let loose from that DNA molecule. There are 2 good animations that I have here for you to look at. This one is on transcription, and this is the one that is included by your book. So your book contains this nice little animation of transcription. And this one is my favorite. This one is animation of transcription in real time. So how fast it actually happens in the cells in your body. It's really, really cool. I highly suggest watching this one because you get to see how fast in just milliseconds transcription can occur on a gene. All right, as I mentioned, before the RNA molecule can actually exit the nucleus, there have to be a few small changes made to that RNA molecule. It's not really done yet. Do you remember when you looked at cellular respiration, how you first go through glycolysis, and in glycolysis, that glucose molecule is being split, and then it goes to enter the mitochondria if there's oxygen present, so that then it can make energy. But once it enters the mitochondria, and before it can enter that Krebs cycle, it actually needs to be groomed into something called citric acid. So it needs to have some addition added to it. This is similar here. While you're not making citric acid, you are still grooming that RNA molecule. You're making changes to it. This is known as RNA modification. And this is a two-step process in which the first thing that you're going to do to that RNA molecule is you're going to add a cap and a tail to it. And this is very important because you don't want the molecule messing up. You know the shoelaces on your shoes? How at the end of the shoelace, they always have that little plasticky tip to it? There's a reason it has a plastic tip. Think about it. If it didn't have the little plastic tip on the ends of the shoelaces, then when you go to put them through the holes, they would mess up. They would tear and break, and then you'd have strands of your shoelace coming apart. So those little caps and tails of your shoelace help protect the integrity of the shoelace. This is exactly similar to the cap and tail added to the mRNA molecule. It's going to help protect the integrity of the mRNA molecule from degradation. There are lots of enzymes and other stuff outside of the nucleus that can break apart that mRNA molecule. And this cap and tail help that from happening. It also helps it from any damage as it exits the nucleus. Okay, so the tail is always added to the 3 end, and the cap is always added to the 5 end. As I mentioned, it facilitates the export of the mRNA molecule and keeps it from getting damaged through the exit process or through the enzymes and acid found outside of the nucleus. It is also that cap, so this end here on the 5 end, is also going to be the part that's going to attach to the ribosome once it finds a ribosome in the cytoplasm. The second thing that's going to be modified on the mRNA molecule is going to be that some pieces of that molecule are actually going to be removed. Okay. This may sound weird because you just made an entire mRNA molecule. Why would you now remove pieces of it? And the reason for that is a gene, one gene can actually code for many proteins. So the same gene that codes for, say, your ability to roll your tongue may also code for the ability to have freckles. I don't know if those are in the same gene, but I'm just giving examples here. So one gene can actually code for multiple types of proteins. And the way that happens is through something called alternative splicing. Okay? This is where if I'm taking what this gene says, but I remove pieces of the gene and put it together, then guess what? Those 3 pieces together code for one thing. But if I have that same gene and instead I'm removing these 3 pieces and then putting them together, then that codes for a completely separate protein. And this is why from one DNA molecule you can make so many proteins. So as you learned in the last chapter, initially people thought proteins were the most important thing and what carried our genetic material and not our DNA because there's so many more proteins than there are DNA. But the reason is alternative splicing, which allows one gene to make many types of proteins. So your mRNA molecule is made from that DNA molecule. And here's an example of an mRNA molecule that was made from a DNA, from the gene of a DNA molecule. These guys in red are called your exons. They're the ones, the blocks that are being cut out and then pushed together to form your final mRNA molecule. So the exons pushed together are your final mRNA molecule. And if you're thinking, how do I remember the difference between exons and introns? Well, the exons are the ones who are finally going to exit the nucleus. Only the mRNA molecule that is finalized with its exons attached is going to exit the nucleus. The introns are the pieces that are left behind when you remove the exons. So your spliceosome is a type of enzyme that actually removes portions called introns, leaving you with a bunch of exons. The exons are pushed together, glued together, and those become your finalized mRNA molecule. All right, the next step after this one, right down here, is going to show you, oh, before we get to that, this is another good image that shows you a DNA molecule making an RNA molecule. So you can see here in red this RNA molecule being made from the DNA molecule. It's only using the gene off of that DNA molecule, so off of the template strand, not the complementary strand. Once the RNA molecule is made, you need to add the cap and the tail to it, and then remove a bunch of pieces called introns, and then take all those exons, you can see exon 1, exon 2, and exon 3, push them together, and there you have your finalized mRNA molecule, which will then exit the nucleus, find a ribosome to attach to, and you will start your next step, which is going to be translation. Before we get started with translation, I'm going to stop the video here to not leave it too long. And in the next video, we're going to look at translation in detail.